Hearing impairment affects millions of people in the world today and can affect a person's ability to relate to others in educational and business settings. The type of hearing loss we are focusing on has to do with the mutation of connexin 26. This mutation is a sensorineural non-syndromic hearing loss that is prelingual. This means the hearing loss is caused by permanent changes in the inner ear before the person can speak. Mutations of connexin 26 is the reason for sensorineural hearing loss. This mutation is passed from parent to child in a recessive manner. Hearing loss starts with damage to the cochlea, which does not allow auditory signals to be made into action potentials and interpreted by the brain. The organ of Corti is a structure in the cochlea of the inner ear that produces nerve impulses in response to sound vibrations.
There are two types of hair cells in the organ of Corti, inner and outer cells. Each have stereocilia that connect to the tectoral membrane. The base of the cells are connected to the basal layer membrane, which when sound comes into the ear causes a shearing force between the membranes and leads to the hair cells to bend back and forth. The bending creates an electrical signal which the auditory nerve transfers to the brain to process as sound. In order to transfer the electrical signal of sound, potassium ions are transported through epithelial and connective gap junction systems. Potassium travels through the sensory cells and eventually gets recycled into the endolymph space of the semicircular canals to be re-entered into the sensory hair cells. Errors in gap junctions make it so no potassium ions get through to conduct electrical signal for hearing.
A gap junction is actually made up of two connexons, and a connexon is made up of 6 connexins. If just one connexin, 26 in this case, is affected, permanent hearing loss is possible, regardless of the ability to replace connexons quickly. This is because connexin 26 is able to make whole connexons by itself, as well as with others, and if they can't be made correctly, then many gap junctions simply aren't available for use. When connexin 26 is encoded incorrectly, either destruction of the protein before it can be utilized or problematic travel to the cell membrane after being synthesized is possible. All of these result in the potassium ion flow being disrupted. The GJB2 gene function is to encode for how to make the connexin 26 protein, and is located on the 13th chromosome and causes the most common hereditary form of hearing loss. One could have a genetic test done on their DNA to determine if they are a carrier of this gene. A common mutation is the encoding for the deletion of a guanine nucleotide, which abruptly ends the synthesizing of the connexin. This goes to show how with just the change of one amino acid, drastic effects can show up in a person's life. In this case, complete hearing loss can happen.